I must say, this is a huge privilege and pleasure to be part of this important meeting. It's an important meeting because it's lifting the veil, hidden no more, in Pope Francis's words, and I think is an important first step to bringing Huntington disease into awareness in Arabic countries and many countries around the world. I do want to say that I'm particularly moved by what Sven Olaf and Astrid are doing around the world to bring awareness, to reduce stigma. There was a meeting in India. And so trying to bring to awareness that this is a disease that is everywhere. And I want to share with you my own personal experience, because when I started working on Huntington disease as a medical student in Cape Town, South Africa, I was told that Huntington disease did not exist in Africa. And as part of my PhD, I traveled to all mental health hospitals in the country and looked to see whether I could find any patients with Huntington disease. And of course, what I found were many patients with Huntington disease in all the mental health hospitals throughout the country. They were called other things. There had been only 2 or 3 publications on Huntington disease in Africa. And the perception was that this exists. At this stage, this disease did not exist. But I then, as part of this was in the late 70s, I was able to go and meet families and saw over 1,000 families in different parts of the country. And here you see, with permission of these families, I was able to take photographs and I'm able to share them. Of course, here's a man with Huntington disease. And I was very moved. I was moved by the courage, the openness, the generosity of spirit, the recognition that this was going to run in these families, and I'll talk about that in a moment. But I was mostly moved by the generosity of spirit and the warmth of these families. And so here I am. You can see it's a long time ago, because you can notice my hairstyle is a little different to what it is now. And these were families that welcomed me, told me their stories. I visited people in the back rooms, examined them, and recognized that Huntington disease was sadly alive and well in Africa. These were people disenfranchised by apartheid and also disenfranchised by this disease, and lived under the stigma and also discrimination that occurred in these families. As part of this, I also met Marjorie Guthrie. And Marjorie, at a meeting in the late 70s, took me under her wing. As you recall, Woody Guthrie died of Huntington disease. And here's Arlo Guthrie, a famous singer in the United States. But Marjorie inspired me to help create the first Huntington disease family organization in South Africa. This was the first multiracial organization in South Africa, crossing boundaries in multiple ways, and that family organization still exists today. So this was very important. We created a multiracial clinic. That was the first one at Groote Schuur Hospital in Cape Town, where the first heart transplant in the world had happened. And here I am with Marjorie. You can see again, that it's been some time. But Marjorie persuaded me at this stage to also consider moving to the United States to continue my work in Huntington disease. But let me say, it was these early imprinting for me, the dignity, the courage, the difficulties that inspired me to dedicate so much of my life to trying to do what we can to alleviate suffering, to bring this out of stigma, and to bring hope to families. And so that's really, it goes back a long way, but I'm really thrilled because I know in Arabic countries, having also interacted with some Arab families with Huntington disease, how this is hidden, the stigma that's associated with inherited mental illness in families. And so this is the first step. to try and bring this to the fore, to let you know this is a disease just like diabetes, just like, sadly, some forms of cancer. It's a disease that has impact in your DNA. And as you know, DNA, here's DNA, it's transcribed into messenger RNA that produces a protein, and this produces a toxic protein that then induces damage in cells and is mostly inherited. I'm going to tell you also that in some families, it's not. So the gene encodes the instructions for this particular protein, and this protein has a particular spelling error, and the spelling error is enhancement of the CAG. The CAG encodes for glutamine. And so when you look at this, this is a polyglutamine disease. It's production of too many polyglutamines that is the cause of this particular illness. We all have the Huntington protein with the normal number of glutamines, which is less than 36. And this particular protein is a very important protein for all of us in the general population. It's neuroprotective. It has very important functions in a cell. But when you overproduce the polyglutamine, this is what causes toxicity, and you also lose the function of the normal protein in this way. So this was the discovery in 1993 by the Huntington's Collaborative Group, where you can see this was a novel gene. This was a big and exciting development. In normal individuals, the CAG is actually less than 36. We now know that. With the Huntington mutation, it's greater than 36. Here it's 40. And here you can see CAG that continues. So this is the gene itself that underlies the basis of this disease. It's important to note that not every patient with Huntington disease has a family history. And in fact, we recognize through a lot of work that about 10, sometimes even up to 15% of patients do not have a family history. And this is important to note because when the CAG is between 30 and 36 repeats, this can expand when transmitted to the germline into the actual mutation that can cause the disease. So it's not always the most, this can arise spontaneously, and it does so everywhere in the world. So here we have it. As you know, it's caused by expansion now of this CAG. It's characterized by some of the findings that Michael and Bernard and Elzbieta has told you about, all the neurological, the cognitive, and the behavioral changes that cause this particular disease in its full manifestation. And as I mentioned to you, patients with this disease who inherited this generally have one Huntington mutation, but they still have a normal gene, where the CAG is within the normal range. Now, what you can see here, this is a gain of function toxicity, and this gene is still functioning normally, but you need both genes to provide some neuroprotection and affect various organelles in the cell to provide normal function. So this is a gain of function associated. And also to note, this is not always inherited. This can occur also spontaneously. So the CAG length, when you look at it, so firstly, less than 27 is unaffected, but there's an intermediate range that can have a risk of expansion. Generally, when you have 36 to 39 repeats, you will develop this disease later, quite a lot of variability of onset, and we recall 40% plus is fully penetrant, where carriers will develop the disease. Now, if you live long enough, and you can develop Huntington disease anywhere from being a child at 2 all the way to greater than 100. And there are patients who are now living longer who have developed this disease at 100. And often these may be patients with reduced penetrance associated with this. So when you look at the age of onset here, and you look at CAG length, what you can see is as the CAG length increases, the age of onset decreases. But important to note And this was really from some of the first manuscripts from our lab and others in 93, 94. What you can see, particularly at lower CAG, there's tremendous variability of age of onset. And Bernard has spoken before about some of the genes associated with this variability, which could be related to somatic instability. But I want to share with you just some findings that really are more specific. And as you recall, and Bernard and Michael have explained this, there's a pre-symptomatic phase where you really are completely asymptomatic. Then there's a prodromal phase where there may be some subtle manifestations associated with a decrease in function. And then finally, you manifest with the disease with some of the classic findings of chorea, cognitive impairment, motor impairments, and also changes in psychiatric findings. The gene actually causes toxicity initially in particular regions of the brain. So here it is. This is the area of early selective degeneration. It's a small area of the brain. And this is important to note because, of course, whenever you're delivering therapy, you want to reach this area and have target engagement. And you can see over time, you get further diffuse atrophy here. You get ventricular dilatation, which is a sign of usually atrophy with tissues are surrounding this. And in the late phases, you get diffuse brain atrophy. The first vulnerability is these neurons, the actual medium spiny neurons in the caudate nucleus that really undergo initial degeneration. So in most patients, if you look at the Huntington disease gene, you have a long CAG associated with some other findings here. These are CAG repeats. And then what you can see, there's CCG repeats. The CAG encodes for glutamine, but this is also, and in most people, you have also a CCG, which also encodes for glutamine. And this is seen there. And the CAA, which is a different DNA structure, but it encodes for polyglutamine. And we've recently, together with other colleagues around the world, have recognized that in some patients and some people, there has been another spelling error where that CAA becomes a CAG, and this then results in a loss of interruption, which is resulting in earlier age of onset. So here we have, again, this particular finding, but here what you can see now what's happened is that CAA, that has changed to a CCAG, and you have a longer tract of uninterrupted CAG. And what we now know is that loss of interruption associated with that is associated with earlier onset. And so some of the variability in onset is influenced genetically by a further loss of interruption. The polyglutamine length is the same, but there is a loss of interruption associated with it. And this results in earlier onset, even up to 20 years earlier in some of the studies. So the symptoms appear earlier with this loss of interruption associated with this. And in fact, what you can see at all CAGs, and this is, for example, this is associated with increased instability. So here you can see the instability, which is seen at all CAGs, but associated with where it's the normal sequence. Here it is the loss of interruption, and you can see there is more instability. Now we still need to show that that instability is what's causing the disease. There may be association with RNA and other approaches, so we still need more data to know that it's the instability that really causes this particular disease. But what we do know is that somatic instability, the instability increases with repeat size, and certainly the instability is associated with earlier onset. And this is a paper from the group in Boston showing an instability is shown by this little trail here. And here you can see the trail, more instability, earlier onset with less instability here. So instability is important, but the mechanism associated with that is still not worked out. We can see instability in the brain, and this goes back to 1994, where we showed even in the caudate in the brain, there is a lot of repeat expansion in the striatum. So the instability in blood is associated with instability in the striatum, perhaps causing more early vulnerability, with less instability in the cerebellum part of the brain, where there is less toxicity and less atrophy. So this effect in terms of age of onset, again, if you have the loss, here's the clinical size, and the age of onset is decreased if you have this loss of the interruption associated with it. So furthermore, if you look at patients who have 36 to 39 repeats, and you say, why would they manifest? Here you can see, for example, at 39 repeats, these are patients who mostly have non-loss of interruption. But as you go lower to patients with 36 and 37 who now manifest as adults, most of them actually have this loss of interruption. So this is associated with early onset. So I think that just explains that there are genetic factors that are associated with early onset. There's the CAG and then the DNA sequence. I do want to say that a lot of this work is done by people in my lab, and here are some of them really from all over the world who are really committed with me to do whatever we can to, including here Amira, whose family comes from Kuwait, and others who, and Egypt. And so we're delighted to play a role in all of this. And I want to say what a privilege it is to participate today in this important meeting. So thank you all very much.